All right, welcome everyone to the Zangwill Club, second year of the COVID, as we usually call it, or 2021 in the old nomenclature. It's really my pleasure to welcome Assistant Professor Emily Coffey. I've been following her since the last maybe 12 years when we met, or even maybe more, when we met in a special course for geeky people about sleep, somewhere in different places in Europe. And at the time, I think she was one of the most interesting people when we were, late students, because she was coming out of training astronauts. And that's also what made her an interesting character, because I think that going from there to the topics she's working now made her, I think, fairly fearless of following what she thinks is interesting. Hopefully, you will get that sense of fearlessness throughout the talk or later in the discussion. I could introduce her in the formal way, but that's already written in the talk, so you can see all of these sort of details about PhDs and postdocs and etc. there. I just wanted to say something more personal. Emily, welcome. If you want to share the screen, and we will start the formal talk.
Okay, thank you very much, Tristan, for the kind introduction and for inviting me. I'm trying to share my screen and it says that I don't have permissions. Maybe
yeah, I'm going to give you only your permission, so there we are.
Okay, let me see. All right, can you see my screen? Okay, so I can keep the chat open in case people have questions, but then I can't go full screen. So I don't know if you usually have some interaction during your talk, or if you prefer that I just
unless there is a very specific clarification questions, we usually keep the long questions for the end.
Okay.
So if I see that something is pressing in the chat, I will probably just, hey, Emily, can you clarify this? I'll tell you.
Sounds good. Okay, and I just see that we have the wrong page, and then when I flip the screens. All right, does that look good? Okay, so thank you very much for inviting me. Today I'm going to be talking about the work that I've done over the past few years, mostly on the auditory system, and now continuing on into sleep research. So as Tristan said, I'm assistant professor at Concordia University in Montreal, Canada, and I'm an adjunct professor at McGill University, where I did my PhD with Robert Auditory, who you'll see features prominently in my work. We still work together quite often because it's fun. So the kinds of questions that I'm looking at are things like, how do we learn complicated auditory skills, and what does it do to our brains when we learn this? And for this, we often use musical experience as a means of looking at experience-dependent plasticity. Of course, there's some debate about how much of that is actually predisposition and genetic or pre-existing differences, and how much of that is caused by training. And that's one of the questions that we're looking at in general, but we won't look at that too much today. But anyway, we're interested in musicianship because it's an activity that's very complicated. It involves multiple brain systems. You can integrate auditory system with motor system, visual system, and of course there's lots of things to do with attention, selective attention, auditory stream segregation and such, which really involve many regions of the brain. And of course there's a lot of overlap with language. So even if you're not interested in musicianship per se, you might instead be interested in other things that we use these systems for. And there's a fair bit of overlap between musician-related plasticity and language learning. So this is kind of the general area. And today what we're going to do is follow a bit of a thread through my experiments over the past, I guess, six years or so that relate to this topic. So I'm going to start off talking a little bit about short-term plasticity in the system when we teach people how to play the piano. We'll go down into the brainstem to look at some very early signals that represent the quality of sound encoding as a starting point for why people might differ and how we can modulate and improve auditory encoding in the system. We'll look at some somewhat more practical applications to do with hearing and noise processing, and what, if you have a better system, what that allows you to do in real life. Then, changing gears a little bit, we're going to talk about sleep and how sound is processed differently across changes in consciousness states, or states of consciousness. And what we could do with this is actually influence the brain using sound to change the brain's ongoing activities that have to do with sleep-dependent memory consolidation and sleep. And finally, we'll bring it back to see if we can try to influence how people learn complex tasks like musicianship by enhancing their sleep-dependent memory consolidation processes using sound. So these are all kind of connected, but yeah, I'll try to draw a thread between them. So I'd like to start off with an experiment in which we trained people who had never played a musical instrument before to play the piano. So the hypothesis was that short-term piano training would cause functional changes in the auditory and motor systems. We also had a secondary question, which was that people would differ in the rates of learning that they had on this complex task, and that we would be able to correlate these learning rate differences with some brain activity before they even started. So this is more getting at the predisposition. So our paradigm was as follows. We scanned people. We weighted in fMRI, so we had also some structural data, but the main focus of this is functional data. We waited 6 weeks. That was just a control to see if anything changed in general. We scanned them again at time 2. Before they had ever played a piano, and then we sent them home with a small MIDI keyboard. It looks like this. I don't know if you can see this very well, but so it's a small portable keyboard, and we had designed an online piano training paradigm such that we could watch what they were doing, and we could check that they were doing their practice and give them a call if not, and say, you haven't done your music practice today, just like you would with your kids. Anyway, they had to practice 5 days a week, half an hour a day, and then we brought them back into the scanner, and we played them music, popular songs like Christmas carols and the Beatles and things like that they all knew. Half of the songs were things that they had actually learned to play now, and half were ones that were familiar, but they had not learned to play them. What we found when we compared the brain activity when they were listening to these sounds at scan 2 before they had learned anything, and scan 3 after 6 weeks of training, is that there was heightened activity in the dorsal stream, so in areas that are connecting up with motor regions, which makes sense because these people were now not only listening but also activating their motor regions when they were thinking about what they had learned. So in this project, we're able to show that it is possible to cause neuroplasticity within the system, even within a short period of time. This is relevant also for potentially for clinical applications, because there have been some other works that show that when you do this kind of short-term training, people's perceptual skills on things like that really matter, like hearing and noise perception in children or in older adults, can actually be enhanced or be improved through this type of musical training. With respect to our second question, we allowed people to progress through the learning curriculum at their own rate. And we found that some people learned so fast they finished our curriculum in the first 4 days, and we had to find them something else to do to keep them busy for the rest of the 6 weeks. Now, other people instead had a very slow learning rate. And even though we knew that they were trying and they were intelligent people and were at McGill and studying, usually undergrads, they had very slow learning rates. So we were quite interested in this difference between people, what you might consider talent if you were watching your children try to learn music, and some of them went much faster than others. When we regressed these learning rates into our brain data, we found that there's a spot in the right auditory cortex whose activity before they learn anything will predict how well they learn. So we hypothesized maybe this is because that's a very basic auditory area, and maybe it's that those people are receiving better incoming information somehow, which is allowing them to do these more advanced things because the quality of the information that area is receiving is just somehow better. So to look at this question, we must go into the system and use some techniques that allow us to look at the finer timescales, finer temporal resolution of the brain's activities. So as I'm sure everyone is aware, the basic way to record an auditory evoked potential is to plug someone into the EEG, play them some sounds repeatedly, average your EEG data, time lock to the sound, and then you end up with something like this, an auditory evoked response. And this has some characteristic waveforms, which depend to some degree on your brain state and on the type of the sound and the amplitude of the sound and such. But this is a basic way that you can characterize one of the things that the brain is doing when you're responding to sound. So we decided to also look in MEG, which has also been done a fair bit, maybe not so much as EEG. And the reason why you might want to use MEG is that you can then do some source localization to figure out where those signals are coming from. And I'm showing this just as an example. These specific results are not so important, but to show that if we select time windows over which we see components that seem to be interesting and seem to correlate with behavior or training, we can then go looking in the brain and figure out where this information is coming from. And the very early components that you see around 50 or 60 milliseconds are coming from the primary auditory cortex. And from there, you see activity arising at secondary and later regions of the cortex and going up to other places like the operculum and then planum temporale and all kinds of things. So we can know from this that these slower later components are reflecting cortical processing. The problem is that the auditory system, of course, doesn't start at the cortex. Unlike the visual system, which also does it, but more so, the auditory system, you have information that's entering very low down in the pontomedullary junction, and then your auditory nerve synapses in the cochlear nucleus, and starts connecting with all different types of cells that have different functional properties. And then you end up with multiple pathways going up the system. And there are multiple nuclei along the way, that each of which has some processing to do and has some specialized function and is treating information in slightly different ways. This is interesting to us because we can still measure what the brainstem is doing using the same sorts of techniques as what I showed before with the auditory evoked response. And if you look at the very early part of the auditory evoked response, you'll see a series of very tiny waves. And these tiny waves are coming from the brainstem, and they represent activity of these nuclei as the information is being passed up the cortex. So we can say that these early or faster components represent subcortical processing. And in this way, we have a way of measuring and quantifying the activity and differences in activity across people and across conditions. Interestingly, there are populations of neurons in the auditory brainstem and thalamus which actually phase lock to the lowest frequency for the most part, the fundamental frequency or the lowest frequency in a complex tone, if it's a periodic sound. So that means if it has multiple waves in it. This is just an image to show that a neuron might not be able to fire at every single cycle, but collectively a population of neurons will fire at a specific phase. Whoops, I pressed a button, sorry. So that collectively they'll be able to represent the frequencies that are actually present in the stimulus. And as you go up the system, the ability of these neurons to phase lock decreases. So at the level of the auditory nerve, you might get neurons firing at a few thousand or being able to represent a few thousand hertz, whereas up in the cortex, it was thought that they are not fast enough to really represent sound in this way. So the three levels of the subcortical auditory system we're interested in are the cochlear nucleus, which is down here, inferior colliculus here, and the medial geniculate body and the thalamus. I'm just pointing these out because I'll be using these acronyms in the future. So how can we measure these things? It's actually really easy. We do the same thing as an auditory evoked response. And in fact, you only need a single channel of EEG to do this. So you get a friend, you plug them in with a single channel to an EEG amplifier, and you play them a sound over and over again. because this is a very small signal with a low signal to noise ratio, you have to do it far more times than you would if you were just looking at that slow cortical response. So say you play this da syllable 2,000 times, then you average it just like you would for an ERP, and you end up with something that looks very similar to the sound wave that you put in at the beginning. This is called a frequency following response, or sometimes it was referred to as a complex auditory brain response. It has two parts. The first one is the onset response, which is here. And that's similar to that first click response we saw in green a few slides ago. And so that's really brainstem as the signal passes up from the auditory nerve up to the cortex. But then we have this beastie, which I'm more interested in, which is called the frequency following response. This is the brain's representation of the steady state or periodic portion of a sound. And it's approximately equivalent to our encoding of pitch perception. There are a lot of caveats to that statement, but that's how you can think about it for now. Just a note on how these are represented. We often do a Fourier transform on the FFR and look at it in the frequency domain or the spectral domain. And this allows us to quantify the amplitude easily. So we'll look at these little spikes. This spike is the brain's representation of the fundamental frequency or the lowest frequency in this sound, which is about 100 hertz. And we can just read off the amplitude here, and we can look at how this varies between people. So why is this interesting? It's because as well as just being an interesting part of the auditory system, it's been found that this scalp recorded EEG relates to all kinds of other things. So people who have dyslexia or language related learning disorders, in older adulthood, your FFR changes. Musicians have higher fidelity and higher amplitudes FFRs. You can actually train people on speech and noise processing and such, and you can see their FFRs increase. People with certain disorders like autism will have different looking FFRs. Anyway, it relates to a whole bunch of different things, even with concussion and language learning. So this is, even though this is a very basic low-level kind of process, it predicts human behavior across a wide range of domains, and that's why it's interesting to us. So I have a little demo here of the ABR, but I think I'm going to play it at the end if anyone's interested, because my computer doesn't like to hop in and out of Zoom or full screen very well. Okay, so we thought that what we would do is record the FFR musicians and non-musicians and see if we could manipulate it or improve it in some way. But we ran into a problem right away, which is that once we started recording people, we saw just how different people's FFRs were. And this didn't make any sense to me, because I said, if we're measuring something that really represents sound encoding, and we find these subtle differences that are relevant for disorders and for experience-dependent plasticity, why, when you look at 5 people's FFRs, they all look completely different? So we did an experiment to explore this, these differences. With the hypothesis that the strength of the fundamental frequency representation in the FFR would be related to perceptual differences. And the psychological phenomenon we chose to address with this is whether or not people perceive the missing fundamental. So this is known as the missing fundamental phenomenon. So if you present someone with a complex tone that has had its fundamental removed, some people are still going to hear the pitch as if the fundamental was still there. And other people are only going to hear the harmonics. And your propensity to hear one or the other can be quantified by playing them a series of tones. So in our first step, we just started looking at individuals' FFRs, and you can see that these are just the time series of 5 random subjects. And some of them are stronger and clear, some are strong but messy, and others are kind of weak. And in fact, you can't just look at the FFR and say that's a musician, this is a non-musician. Subject number 5 was actually quite an experienced musician. So these differences that you read about in papers, they work out on average, but there's still a huge amount of variability between people. So we looked in the spectral domain and we quantified people's FO amplitude, and then we related it to their perception. So how likely they were to hear a missing fundamental tone that wasn't there. So here we've got the strength of the FO on the y-axis and the perceptual bias on the x-axis. And we can see that people who were more likely to perceive that missing fundamental are those who have the stronger FO in their frequency following response. Note that the signal was not actually there. The brain is representing something that's at least not in the original stimulus. Yeah, I think I'll leave it there because it's actually in the modulation of the amplitude of the signal, but that seems to be what the FFR is picking up on. So that got us curious, didn't really solve anything, but it got us curious. We were wondering, where do these differences arise? The experiments I've talked about so far are only using single channel data, and you can't tell where that's coming from. So we thought, well, if you can look at these slower responses using MEG and figure out where they came from, why don't we do the same thing with MEG? So we put 20 people in MEG. We had them listen to a repeated DA stimulus for an hour. We got 12,000 iterations of the DA, and then we tried to localize it in the MEG data using their own MRI-based physiology. Sure enough, we were able to isolate signals coming from the cochlear nucleus, inferior colliculus, and the medial genicular body of the thalamus, with somewhat different amplitudes, but clearly above the noise level. But we also had a surprise, which was that there was a strong signal coming from the right auditory cortex. We didn't think this was possible because the FFR has been interpreted until now as being coming from the brainstem because of those phase locking limitations in the neurons at the level of the cortex. So we wondered, does this have anything to do with behavioral skills in these subjects? So we got the strength of the fundamental from both the left and the right auditory cortices for each individual. And we first tried to relate them to the number of musical training hours in our sample. This was a kind of a convenience sample with respect to musicianship. So we had a fair range. And we found that on the right, but not in the left auditory cortex, the strength of the fundamental increases with the number of training hours they've had. And conversely, it's stronger for people who had a very early start to their training. We also looked at some perceptual skills, and we looked at frequency discrimination. So you hear two tones and you make them closer and closer together and see when people can tell the difference. And people who have a stronger fundamental frequency in the right auditory cortex are better at this task. So we have a new interpretation of the FFR, and we have a new tool, MEG, for looking at the system as a whole, being able to break it apart and look in the whole human system non-invasively at what some of these different parts are doing. And there are some implications of this work of there being a cortical contribution. So there have been many, many studies on the FFR and its relationship to all of the factors I talked about before. But it's possible that many of these effects are actually only happening at one level or are happening in the system as a whole and are not necessarily constrained to the brainstem, which has been the common interpretation. So we had a few problems with reviewer 2. So I'd just like to point out here that 5 other groups have now replicated our results here using EG, MEG, and also intracranial recording. So we're quite sure, at least within the low frequency range, there's a contribution from the cortex. The cortex does have phase locking limitations, of course. I expect they're around 200, 250 hertz. Nonetheless, the FFR that you record to these lower frequencies still seems to predict performance on a range of other behaviors, even that are using the full range of frequencies. So for some reason, it's very predictive, even though you're looking at a very small range. Anyway, so coming back to that first study, we saw that those people who learned faster seemed to have more activity in the right auditory cortex before they even started. And we thought, maybe greater bold responses in the auditory cortex are related to stronger FFR, better sound encoding. So we ran another study looking at the relationship between the FFR recorded outside of the scanner, because we didn't have an amplifier that would go to high enough sampling frequency inside the scanner, and in the same subjects inside the scanner. And our hypothesis was that the bold activity in the auditory cortex, especially on the right side, would be related to the amplitude of the FFR recorded in EEG. We're also interested in the distinction between the functions of the right and left auditory cortex. So I put this in brackets because it's a bit of an aside, but we're expecting this pitch processing to mostly be in the right hemisphere, and conversely, we expect fine temporal processing to take place in the left hemisphere. And we do actually have a measure of fine temporal processing, which is that little onset response that you saw at the beginning of the ERP. So the logic is as follows. If you have a stronger FFR, then you have more neurons that are firing in a more synchronized manner, and those will have metabolic requirements. So you'll see a stronger bold response for those people who have a stronger FFR. So recorded EEG and fMRI in 27 people. They listened to about 2,000 repetitions of this speech syllable da. And we also wanted to replicate this in another kind of sound that's similar. So we used a piano tone. It's a G2 tone, which also has a fundamental frequency around 100 hertz. And when we looked in the right and left auditory cortices, we found that there were regions in it looks like the plenum temporale here, which are related to the strength of the FFR. This was true for the whole group. It was also more true in musicians. They were the ones that had the strongest ones. And it was replicated across speech and piano tones. We also overlaid our previous MEG result on it to show that it's ending up kind of in the same region, and making some allowances for these being very different signals, of course. We then also looked at the timing of this onset response that's happening at the beginning, and we found that it correlated with the left auditory cortex activity. So we think this might be looking at the beginning of sound features and their processing, separating into right auditory cortex for fine pitch processing and left auditory cortex for fine temporal processing. So then we wondered, well, how much of this is burnt in and maybe predispositional or something that's really hardwired and you can't change, and how much is something that you can change online just by wanting to, or by paying attention to something. Now, we do have strong reasons for thinking this is a case, and one of them is in the structure of the system itself. We have many more descending connections in the system than ascending connections, and that makes a kind of a substrate for top-down modulation and even neuroplasticity over longer timescales. So in this experiment, it was a follow-up to the previous one about the fundamental. We asked people to hear the missing fundamental. It turns out that if you choose your stimuli carefully for each individual, you can find a kind of ambiguous point at which they can hear it either in the fundamental mode or in the spectral mode. So we had a whole procedure for finding just the right harmonic series to use for each individual. And then we would construct a melody for them out of those stimuli. And we had a few primes that were just pure tones, which told them what the melody that they were supposed to be listening for should sound like. And those were either based on the fundamental or the spectral perception. And then we had them just listen to these missing fundamental melodies over and over again. And we said, okay, listen for the fundamental side, listen for the spectral side. And our hypothesis was that the strength of the fundamental frequency would be related to whether you're trying to hear the fundamental melody or the spectral melody. I should have shown this. This is a representation of the tone itself. So here's the fundamental, or the lowest frequency at 98 hertz. These are integer multiples, so the second, third, fourth, etc. harmonic. And for, say, one individual, the 8th, 9th, and 10th harmonics sound a bit ambiguous to them. They can hear it both ways. For another person, you might need to hear the 2nd, 3rd, and 4th harmonics, and they would be able to hear that both ways. So we found that the neural representation was modulated by the attentional condition, not always in an intuitive way. It would actually kind of depend on the person. It would be consistent within person, but differ between people. Anyway, we took from this that top-down effects on early representation are possible, and you can actually manipulate the FFR just with your attention. This is work from another group. This is from Thomas Hardman and Nathan Weitz in Salzburg. And they did a follow-up study using MEG FFR, and they had a manipulation in which people were asked to attend to an auditory stimulus or a visual stimulus. And they looked across the whole system as we did, but they found that modulation according to attention only happened in the right auditory cortex. So that seems to fit pretty well with our previous results, showing that a lot of the modulation is actually at the level of the cortex and not necessarily at the lower levels. So we were wondering, we wanted to push this a little bit further and look at how does online enhancement of sound encoding help us with stream segregation. So now we're talking about separating sounds out of noisy environments, things we want to do in real life. And this experiment was inspired by a visit to the grocery store. So I was doing my shopping and I heard background noises, people talking, just grocery store noises. And then at some point, I suddenly heard a song that I was familiar with on the radio. And from then on, for the next 5 minutes, as that song played, I was very, very aware of that song, and I could hear the words and the melody very, very clearly. Whereas before, I had not even been aware that music was happening somewhere in the background with all that other noise. So this had me intrigued. So we came up with a paradigm based on this, and our hypothesis was that we wanted to measure both these fast responses that are associated with the frequency following response, and also these slower modulations that are associated with syllable rate and tempo, or the beat in a tune. And our hypothesis was that the later, slower indices of sound representation would be more affected by attention than the earlier, faster ones. So we ask people to follow the melody. This is a little bit hard to describe without a demo. So at the end, I can play what the stimulus actually sounded like if you're interested. We had a musical excerpt, and it was kind of Guitar Hero-like scrolling representation. And we provided people with visual, spatial, and predictive cues. Predictive because they knew the song, they heard it a few times first. Visual because they had this kind of scrolling representation, and the note that was being played would be highlighted in white. And also spatial information. We made the sound they were supposed to be attending to louder in one ear, and the background sounds louder in the other ear. And this gave the impression of some kind of spatial separation. And then we took the EG that we recorded at the same time, and we filtered it in these two frequency bands we're interested in. So for the amplitude envelope responses, that's about 1 to 9 hertz or 2 to 9 hertz. And for the pitch or FFR related frequencies, we filtered it from about 80 to 300 hertz. We then used a machine learning algorithm to try to reconstruct the audio from the EEG. And the measure you get out of this is the audio you've tried to reconstruct versus the audio that was actually played. And you get a kind of an R value, a correlation value. So the reconstruction correlations are on the y-axis. And here are the different conditions we had. First, we tried to reconstruct the entire sound stream. So everything that was going on at the same time. I forgot to mention that it wasn't just one sound stream. We had it embedded in many different other versions of the sound stream. That was very confusing. It was like a whole cacophony of sound. So it turns out that we're able to reconstruct the high frequency of the entire stream very well, but the attended stream only a tiny little bit above chance. Whereas if you look at these low frequencies, we find that you're not even able to construct the entire sound stream above chance. But if you look at the attended stream, that's now being enhanced and strengthened and is represented much more than anything else that's going on in the sound stream. So we take this to mean that there's a kind of a progression of enhancement as you're going up the auditory system and as you're going to later processes. I think that's quite consistent with some of the other results by groups like Malmiarca in Spain, who've looked at this kind of progressive refinement of auditory information over time and over the system. In our next experiment, we're looking at we're wondering, is there a neural mechanism to enhance useful periodic information? So this is new work, and I think it might be a little bit esoteric for some viewers. So I'm just going to do it quickly, just to give you a taste for those who are interested. We're looking if there's an oscillatory entrainment of the frequency following response. So this has been found at the lower frequencies already, that there's some kind of entrainment of these slow frequencies. We wanted to know, is there something going on also in these FFR frequencies? So we played people DAWs again, and we filtered the audio. This is just a control to make sure that our filtering is not introducing extra oscillations. And then we looked at our frequency follow response from the cortex and from all the other regions as well. And we found that especially at the cortex, we find a very strong and long after effect. So there's some kind of oscillatory activity that's continuing in time. And it doesn't seem just to be the delays incurred by this feed forward system of kind of slowly passing information up to the cortex. One of the reasons we think that is because if we do a sliding window analysis to see how the frequency content changes in time, especially at the auditory cortex, we see that the frequencies actually converge over time. So the stimulus is played for this whole time, but the brain starts off representing something down around 80 hertz. And as time passes, it seems to accrue information concerning the pitch that it needs to represent. And after about 100 milliseconds, then it's able to accurately represent the pitch at the cortical level. At the subcortical levels, we see instead a much higher fit, more like the stimulus frequency even from the beginning. But you see this after effect throughout the system. Anyway, that's if anyone's interested in that, we can talk about that more later. I don't want to get too deep into the little mechanics of the auditory system here. But what we do want to do is look at whether the FFR strength relates to advantages in hearing and noise perception, because that's a real human activity that causes a lot of trouble to certain individuals and can affect children's education, it can affect social isolation in older adulthood, etc. So we did an experiment to look at whether the strength of the FFR is related to speech and noise perception on a separate task. Importantly, in this experiment, the FFR is recorded passively. They're not doing anything. They're distracted away from the sound by seeing a silent video. And the measure of speech and noise perception we have is completely separate. We're using the hearing and noise task, which gives people sentences and noise and asks them just to repeat back whatever they understand. So these things are very separated and are happening at different times. Why this is interesting, more interesting, we think, than doing it at the same time, is you're looking at the system's basic recording without any kind of top-down influence, and how that relates to a very complex, very separate task recorded at another time. Nonetheless, we're able to find a positive correlation between the strength of the FFR, once again at the right auditory cortex, but this time all throughout the system, in that people who have stronger FFRs are those who show a higher speech and noise accuracy. We furthermore showed that the age that musical training started was related to the skill, suggesting some kind of neuroplasticity was involved. People like Nina Kraus, Samira Anderson, and others have been looking at how music can be used as a potential strategy for remediating speech and noise deficits in aging and in younger people as well. Already mentioned about this passive task. So this was a few years ago now, about 4 years ago now. And at that time, people were really debating whether or not there was actually an advantage among musicians for this task of speech and noise perception. We had a lot of trouble writing up our introduction and our results section because it was, we kept having to say, oh, these studies find yes, these studies find no, but all of the paradigms they were using were extremely different. So we thought we'd look at the literature at that time on speech and noise processing advantages in musicianship and try to organize it in some way. We ended up with this. So the main way that these paradigms varied is in the target properties. So the thing that you're listening to, those could be just very basic things like tones and phonemes. They could be hold words or sentences, increasing in complexity in this way. and also the qualities of the background noise. So some of them were just presented in quiet, so no noise at all. Broadband noise, which is energetic but not informational masking. And then up here, you could also do something like we did in our grocery store study when we have multiple streams of music or of speech that they're having to pick out the information they need from. A few observations. Oh, so red are conditions in which they found a significant advantage amongst musicians as compared with non-musicians, and black is the ones that they didn't. So one thing we can see about this chart is that down in this corner, in which you're presenting very simple stimuli to people in quiet or in just some broadband noise, you almost always see a musician advantage. Whereas if you look in this quadrant, it's much more mixed. And we interpret this as when you present people with whole sentences, they have lots of redundant information that they can use. And people can have different strategies, and some people might be better at predicting the syntax or other aspects of language. So you just end up with more variability. But overall, we think there's actually a fair bit of evidence showing that musicians have an advantage on this task. So we wanted to go a little bit further with this. One thing we noticed in this review is that there are very few studies on how musicians might be able to use some of these cues better than others, or better than other people. There are only a couple of these tasks that are using the visual system, yet we know that that's the first thing we use if we go into a crowded restaurant, back when we could do that, and we're sitting with our friends at the table, we're trying to hear them above the music. The first thing you do, if you can't hear them very well, is look at their lips and look at their faces. So the visual cues are extremely useful to us in this task. And we wondered, are musicians better at using visual cues? So we designed another experiment looking at using fMRI, in which we had the hypothesis that musicians would perform better and would benefit more from congruent visual information than non-musicians would. Our paradigm was as follows. We're still using these little images that were sliding along and highlighting the notes that they heard. In this case, they had to learn 4 melodies in advance, and then they were presented embedded in informational noise masking. So that's this cacophony of other musical noises. And we varied whether the visual information they received was congruent. That means it was helpful and it showed the notes that they were actually listening to, or it was incongruent, so they got the wrong one. And we asked the question, is where in the brain are, first, where in the brain is there more activity during the congruent versus the incongruent trials across our whole group? And we find that these non-primary visual areas and multimodal areas are recruited when you have useful visual information. And when you do have useful visual information, conversely, you're less reliant on the auditory regions and maybe some things to do with selective attention and working memory in the frontal regions. We had a group that was made up of half musicians and half non-musicians. So then we looked to see if the musicians were doing something special in this same congruent versus non-congruent condition. And we found that that does seem to be the case. These musicians are more strongly recruiting non-primary visual and multimodal integration areas. So we think that musicians are better at using visual information on auditory tasks specifically, probably not just on visual motor or some other kind of multimodal integration. So changing topics a little bit, now that we know something about how the auditory system can change and how it's reacting in different frequency bands, we were curious how this changes over different states of consciousness. And one of the reasons for looking at this is very practical. FFR has often been recorded when people are encouraged to drowse or to fall asleep, because it's a very small signal, and the stiller and more relaxed they are, the better the recording is going to be. So people say, okay, well, either watch this film or close your eyes and relax a bit, and we'll record FFR because the brain just does that anyway. And we wanted to know, is that really the case, that the brain just keeps doing the same thing? So there's very little recent work on this. There's a bit of work from the 90s, which seemed to say that there's no difference in sleep versus wakefulness. And that's the basis for which people are allowing their subjects to fall asleep. But there was one recent study by Mayan colleagues in Neuroimage 2019, which showed that when people were just allowed to drowse, those who were more drowsy seemed to have a decreased amplitude of FFR, suggesting that the state of consciousness was in fact important to the sound encoding. They didn't have a full sleep study. They just had people who were drowsing a little bit. And the way they quantified drowsiness was how many sleep spindles they were able to detect. So when you fall asleep, when you go into non-REM sleep, some of the first indices that appear that you're actually into stage 2 sleep are sleep spindles, and later on slow oscillations. And sleep spindles are also thought to potentially play a role in blocking sound. So we thought we'd put people back in the scanner, would have them listen to ... again, and would watch as they fell asleep, and then would separate the FFRs depending on what stage the individual was in when that sound arrived. And we found, when we looked across the whole system again, we found that the amplitude of the FFRs in the right auditory cortex changed a lot with sleep stage. So it decreases in amplitude as you go into deep sleep. Whereas the amplitude of the FFR in the subcortical structures didn't change at all, or at least not significantly. So this is work that's still in preparation. Our next steps that we're doing right now are also picking out smaller events within sleep, like these slow oscillations and the sleep spindles, which are things that occur characteristically during sleep, which are related to memory processing and sleep-dependent memory consolidation as well. So we're interested to know if we can influence them with sound, and also what happens to sound processing when these sounds enter when the brain is doing something else. So more to come on that one day. And of course, we can also look at slower responses in sleep. And this is something that there was a paper in Neuron in 2013 by Jan Born's group, who discovered this thing called closed loop auditory stimulation, or they discovered its functional role, I should say. So what happens is you put someone to bed, you record EEG, and you have a computer which takes that EEG, filters it in real time, detects a slow oscillation, and outputs a sound at a precise phase in relation to what the brain is doing by itself. So here, you would want to detect a slow oscillation peak and present sound at this time, this time, this time. It doesn't really matter which sound you use. People have mostly used clicks and 50 millisecond pink noise bursts, and that's probably because you just want to send a little impulse of information kind of up the auditory system. But anyway, the interesting part is that this affects sleep-dependent memory consolidation. So this paper was so interesting because once they did this auditory closed-loop stimulation, they found that it would boost subsequent slow oscillations, and also, not shown here, they would get more sleep spindles associated with this sound being played. And then they taught people very simple tasks before sleep. They did this to them during sleep. When they woke up the next morning, their performance was increased. So they concluded that we're actually stimulating these memory consolidation mechanisms with sound in such a way that we can enhance performance and enhance learning. This is replicated, this is more like 20 times now, this is, needs to be updated, but we don't really have any idea how this works. So we thought, well, maybe with our tools that can get at some of these things in the auditory system, maybe we can try to understand the mechanism a bit more of how auditory closed loop stimulation is actually stimulating memory consolidation. So our hypothesis is that deeper sleep will result actually in higher amplitude responses. This comes from a bunch of older ERP literature showing that when people fall asleep and go into deep sleep, you actually get the regular components, the first components we normally look at that occur within the first few hundred milliseconds of a sound being played will maybe diminish a bit or stay the same. But then as you fall asleep and go into deep sleep, you end up with a really strong reaction at about 350 or 550 milliseconds post-stimulus offset. That looks completely different to what happens during wakefulness. So based on that, we sent them back into the scanner. This time we're presenting sounds with more space in between. Normally when we do our FFR work, we want to present our stimuli at about 5 hertz, so dot, 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 to get as many as we can. Here, since we're looking at these slower cortical responses, we want to see their entire evolution. So we give them give the brain about 5 seconds in between to go back to what it was doing before. When we make our ERPs based on this, we see these first components that we're used to looking at in wakefulness. Wake is in green. Then we have descending into deeper sleep in blue, purple, and sort of pinky purple. And we do see these components that have been reported before at about 350 milliseconds and about 550 milliseconds. Interestingly, in very deep sleep, that's N3 or stage 3 sleep, you end up with this great big oscillation that happens and looks very much like a slow oscillation that happens over the next 2 seconds or 2.5 seconds. And of course, using MEG, we're able to trace this back to the brain and figure out where these things are coming from. So we first looked in the orange box, that's the 350 millisecond component. And we can see that while there is some activity happening in the auditory cortex, which is a little bit hidden here, we also have some activity in the orbital frontal regions, so on the ventral side of the frontal regions. If you go along a bit and look at this later component of 550 milliseconds, then we see that the activity in the auditory cortex has somewhat died down or moved a little bit posteriorly to the planum temporale, but you see a very strong response from now the frontal regions. So we think what's happening, well, one thing that we're going to be looking at in the next little while is try to see how this signal propagates. Is it going up to the thalamus and then causing some, or the brainstem and causing some kind of general alerting response, which is stimulating this slow oscillation process? Or is it going up to the cortex and then kind of leaking out of there and affecting the slow oscillations and the sensitivity of the tissue in that region that's then sort of propagating around the brain? We're not sure quite what's happening in there yet. A nice thing that we can do in this MEG analysis is we can go in and look at the activity in specific regions. So we can kind of explore what's going on. So if we take this here, which is the right gyrus rectus, we can go and make a similar plot to this. And we see that there's almost nothing in those first auditory components. It's not an auditory region, so it makes sense. But here we see this slow oscillation developing that could possibly have something to do with auditory closed loop stimulation. Whereas if you look at the right anterior transverse temporal gyrus, or the primary auditory cortex, you don't see so much of this slow oscillation generation, at least not so much as here, but you do see these initial components. So we're still working on this. It's been a bit slow, but we're hoping to get something out on this relatively soon after some more conductivity analysis. We can also explore what happens when sounds enter the system when different phases of the slow oscillation are going on. I think I'm going to skip over this a little bit. Okay, so our final question is, can we boost complex learning? I should say complex learning with closed loop auditory stimulation. So going back to musical training, we want to know if we can influence not just these really simple tasks that have been studied, like generate a list of words and then a second list of words and try to associate them together and see if people remember more the next morning. We're interested in real human tasks, like these things that we do with our entire systems at once that involve multiple brain regions, etc., like music and like language. So we're developing, well, we're just starting an experiment now in which we're trying to modulate brain activity in sleep, and we're letting people learn the piano before and be tested after their sleep, and we're trying to see which aspects of piano learning can be increased with auditory closed loop stimulation. Our hypotheses are that closed-loop auditory stimulation after piano learning will result in better performance, probably on specific aspects of the performance, like the timing of the movements. We also think that there will be quite some differences in responsiveness to sound, and that these will predict performance gains. So those that are stimulated by sound and have a large brain reaction are going to be those that learn the best. Of course, if you're looking at individual differences, we need a much larger sample. So what we're doing is a large-scale study. We'll be looking at about 200 participants. We're collecting data in their homes, and we'll be doing multiple learning paradigms on different nights so that we can compare them, including some of these earlier simple paradigms that have been shown to work with auditory closed-loop stimulation. We'll be making EEG recordings with a headband that has dry electrodes in it, and we'll be doing auditory closed-loop stimulation in half of our subjects. My postdoc, who's Puya, who's working on this, is very interested in genetics and has done a bunch of that work as well. So we'll be collecting saliva samples to see why people differ in their responses to sound and in the effectiveness of closed-loop auditory stimulation. We will also put this all in an open access archive so that other people can get other things out of it. So we've planned that in from the start. Okay, so that brings us to the end of my talk. Just a quick recap. We started off wondering how we can change the brain with experience-dependent learning. And then we looked down in the brainstem, and the deep structures and the early encoding. We connected that up with some real complex tasks like speech and noise processing and how musicians have advantages on it. Then we looked across brain states to see how processing changes in the auditory system when our brain is doing other things. And then we looked at how we can use our knowledge of the auditory system to actually manipulate memory and processes of neuroplasticity. And then we bring that back to try to improve people's learning on a complex task on musicianship. So I'd like to thank all of my trainees and collaborators, and also thank you for your attention. And I'd also like to point out that we have a few open positions, PhD and 2 postdoc positions, if you have any graduating trainees that might be interested in joining us. And very finally, this is what we've been doing for the past 3 months. We're having this discussion at the beginning of the talk about quincies. So I wanted to add a picture quickly to show you what they are. So this is a Canadian snow cave, and we've been spending a lot of time in our backyard making really fantastic ones. Anyway, thank you very much for your attention. I'm going to stop the screen share.
Thank you very much, and you can join me in clapping or congratulating Emily in however you can or want. Thanks very much. Almost all the papers that you were referring to, I put it in the chat, except of course the ones that are in preparation, so the people can sort of click and see the details if you want to geek out about it. Of course, there are all in the Google Scholar if you find them, but I thought it was good to have everyone on board about the details. You can ask questions by unmuting yourself and asking the question. You can ask questions in the chat, and you can ask questions in the chat, but without telling everyone, just to me, and then I'll ask the question anonymously if you're that shy. And there's no problem in any of these situations. So I open the virtual floor for comments, questions, and discussions. Who wants to start? I think Irvin and Maria are sort of waving in my screen. Go for it. Unmute yourself and ask the question to start.
Shall I go first?
You go, Maria, and then it will be.
Thank you, Emily, for a really fascinating talk. I have lots of questions, but I'll choose one to ask now. And so it has to do with attenuation of the FFR during sleep that you were talking about. To what extent do we know, it also relates to the individual differences in the FFR that you were showing in the very beginning of the talk. I mean, to what extent do we know whether these are real differences or real attenuation during sleep as opposed to just noise? Brain noise or individual subject noise in the case of the individual differences.
Well, it seems to be relatively statistically robust. I guess replication is the best way of answering that question, potentially in a larger sample size. We're also able to see that within subject, the statistics works out. I don't think we've actually finished preparing those results for showing, but it seems consistent enough. So yeah.
What I mean is, I guess, like not noise in the statistical sense, but like, you know, real brain noise. I mean, so we know, right, that brain activity changes, for example, during sleep in quite profound ways. So would that just mask a signal that is present and potentially, you know, equally strong in the brainstem in a way that will prevent you from seeing it or will make you conclude that it's attenuated, as opposed to it really being attenuated?
Because we're looking at a very precise frequency range, we know exactly where the frequency will be. It should be around 98 hertz, which is the stimulation frequency. You don't get huge changes around that frequency range over sleep stages. And in any case, we calculate our amplitude relative to baseline that immediately precedes the stimulus. So that should normalize a bit or get rid of the effect of changing background noise. But I think it's less of an issue within this frequency range. I think if you're looking at the slower responses, that might be more of an issue because you get that 1/f sort of background noise changes that are quite pronounced. I think we should be able to follow up with our next steps for that project. We're looking at sleep microarchitecture and what happens to the FFR if you happen to hit a slow oscillation in its peak or trough, or even if you happen to hit a spindle in its peak or trough, or the refractory period after a spindle. So in part, investigating the microarchitecture of sleep with respect to incoming sound, that might help to answer that question a bit.
Cool, thank you.
Awesome. I think Brian wants to talk or ask a question.
Yeah, you talked quite a bit about the relationship between musical training and speech in noise perception, and you speculated that maybe musical training could help to overcome some of the bad effects of increasing age, for example, on speech in noise perception. But why do you think training with music would have a better effect than training with speech? You know, what extra do you get from music that you don't get from listening to speech anyway?
I think this is a really interesting question. Some of the reasons that have been proposed for this to answer this question are that the musical system is really engaging the reward network. And it could be that that's increasing neuroplasticity. It could also be that it's a supplemental activity. We're all speaking all day long for the most part. But if you go off and then do another activity that's requiring you to focus your attention specifically on the pitch aspects or specific aspects of vocal production through that feedback loop, then perhaps that additional attention and the rewards maybe together are going to increase neuroplasticity. So there are a couple of studies on that. People working on that are Frank Russo in Toronto, I think, Nina Kraus at Northwestern, and Samira Anderson, I'm not sure where she is, but they've all done short-term training studies and shown that the FFR can be increased in amplitude in either older adults or children, and that there's an improvement on the speech and noise perception. So I'm not really sure why the music is so good, but maybe it has something to do with the reward value.
Emily, can you see the chat? There are a couple of questions there, one from Joao Araújo and the other one from Ian Smith.
Okay, let me see. So maybe I'll just read it out. So from Joao, thank you for your talk. You're welcome. I'm curious on how you are going to choose the auditory stimulus properties for understanding how closed-loop auditory stimulation may affect music learning. Are there any specific auditory stimulus properties known to influence music learning rates the best? So we don't know yet. We made a big table of all the studies that have been done on auditory closed-loop stimulation so far, and almost all of them are using either a click or a 50 millisecond pink noise. So we think that's just like a kind of little impulse that you're sending into the system. We've mostly been working with this DAW stimulus because we want to compare the vowel portion, the periodic portion across brain states and such. So we could just use that. But what's been the only thing really that's been done so far is this click or small pink noise. I think there's one study using complex tones, but it wasn't systematically comparing complex tones with other other types of stimulations. We actually don't know what's best. I suspect it's kind of just the sudden impulse. There's also a group that's working on something called targeted memory reactivation. And in this, they're trying to associate things that people are learning with specific sounds. And then they play those sounds selectively to different people in the night. And they try to reactivate memories through this association. And they're combining this also with auditory closed loop stimulation to see if they can selectively enhance learning on specific tasks. But we don't know how well that works yet. They haven't done the recordings. Okay, there's another question in the chat. Do you think your musicians choose to be musicians because of these skills or learn the skills through rehearsal and repetition? So I think it's a bit of both. There are a couple of nice reviews on that, sort of arguing which aspects might be attributed to predisposition versus training. We do have strong reasons to think that both are true. So if you have someone who has a poor auditory system for whatever reason, it's just not encoding some aspect of sound very well, they're likely to find musical training difficult and they won't progress very well at it. Most people would, in childhood, would probably give up or choose another activity. They wouldn't get much reward value also out of continuing their musical training if they're worse at it than all their friends. So there's definitely an element of self-selection. However, we can see through this handful of longitudinal studies that's being done that you can actually affect neuroplasticity in adulthood even through these kinds of interventions, so that we know our systems remain plastic to some degree throughout our lives, and we can use this to enhance our system. So what degree is it one or the other for an individual? It's really hard to say, I guess. Yeah.
Awesome, thanks. Any more verbal or sort of video questions there? Otherwise, I have a bunch, but I think Maria has also quite a lot in her list. But I encourage anyone to just ask any questions. I think Lorena is coming in.
Yes, had you look what happened in REM? In REM sleep, because you have talking about the slow waves and not the.
Yeah,
we
haven't for practical reasons. So we've convinced people to sleep in the scanner, but if you try to give them sounds presented at a high enough sound level that you get a nice FFR for the whole night, they just sleep really poorly. You can get them to sleep for a couple of hours, but that will, and we usually do this in an afternoon nap format, and they do sleep well enough, and you can get the first 3 sleep stages, but we haven't gotten any REM in the scanner with an afternoon nap. We have done whole night recordings, and we've got a little bit of REM in a couple people, but it's not that comfortable to sleep in the scanner for the whole night.
Yeah, that makes sense.
If we wanted to do that, though, we could have them sleep nearby and come in for the second half of the night, but you might have to do a comparison between nights within individuals, one for the slow wave sleep and one for the REM sleep. We haven't tried it yet.
Okay, thank you.
I go? Shall I go?
Yeah, go.
Sorry, I want to ask about this, what you called oscillation. So this sort of slow evoked response that you saw in your sleep data. So you referred to it as an oscillation, but at the same time, it was also evoked by the sound, or at least appeared to be. So I wanted to ask kind of 3 related questions in the sense of, is it really an oscillation?
Sorry, are you referring to the frequency following response entrainment, or the other one?
No, these were your evoked data. So these were the data that you showed towards the end of the talk. Yeah, so I was curious about this one. Is it really, so does it only appear to be, well, clearly it is evoked, I guess, because it's time locked to the stimulus. So
yes.
What could it be? And is it really an oscillation?
Yeah, I mean, the term oscillation is not precisely defined, I guess, because you could have an oscillation that's just, a slow oscillation by definition can even just be a single wave, right? But it doesn't mean that it's something that repeats periodically. So I guess it depends a bit on the definition. I wouldn't think that this is an entrainment type mechanism. You only get one wave or two waves afterwards. And it seems like that's a bit hard to sort out because you also have damping mechanisms in the system. So you don't want the brain, when it's exposed to an impulse of sound, to just continue doing that forever. You need to have a network that will sort of dampen that out and decrease it over time. Hongi and Go and colleagues also did a follow-up study to see if they could increase slow oscillations in deep sleep by presenting a series of clicks to kind of continually boost these responses. And they found that they didn't get any benefit for that. So whether we interpret that as you only getting a series of evoked responses, or that there's a self-limiting or dampening mechanism in something that is actually oscillatory, it's impossible to know from that. So I know it's kind of a non-answer, but I'm much more confident about there being an oscillation for the end of the frequency following response than I am for this kind of evoked response in the slow frequency band.
You go, Bob.
Yes, could I just clarify that? That response which you saw continuing after the stimulus, was that phase locked to the F0 of the sound?
This is the weird thing. So in the lower levels, if you're looking at the cochlear nucleus inferior colliculus, it does seem to be the case. It's representing the frequency content very accurately. But as you go up the system, if you end up in the auditory cortex, the frequency that it starts with is not the same as the frequency that you're putting into the system.
Right.
So it's not just a feed forward thing that's kind of going up the system and that's the frequency. So what we're seeing instead is that the frequency actually converges over time. And it seems to require multiple impulses of information at each cycle to kind of entrain or to get to the frequency that you're trying to represent, which is a bit of a surprise, but suggests some kind of entrainment.
Yeah, so the thing which went on after, so you had a trace which went on for about 100 or 150 milliseconds after the offset of the stimulus.
It's closer to about 40 milliseconds, actually.
40 milliseconds, okay.
Yeah.
And that's and that was from the right auditory cortex.
Yes, so we see it in the left as well, but it's stronger in the right. We also see continued activity in the lower regions, but we don't see the same pattern of convergence and divergence of the stimulus frequencies. But
for that 40 milliseconds after the stimulus had stopped, was that phase locking at the F0 of the stimulus or at some other rate?
So it gets to the F0, and then maybe I could just show this screen again. Just 1 second. Share screen. Oops. You see this this picture?
Yeah.
Yeah. Okay. So what we found, we did this sort of sliding window analysis to see how the frequency content in the FFR changes over time, and we see that so the stimulus should be here. That's at 98 hertz, but instead we find it down here at 80, and then it progressively entrains. Now, once the stimulus stops, it seems to wind back down again, like it's kind of relaxing to a I don't know, previous state.
Yeah.
And we see this in the left and right auditory cortex, much less so in the lower regions. We have a lower SNR in the lower regions, so I don't know if it's just noisier or it seems to be that it's following the stimulus more veridically.
Yeah. I mean, did you have something on the previous slide to that which showed something?
Yeah, that's just the after effect.
Yeah, here. Yeah, the MEG FFR right auditory cortex. So what I'm thinking of is that if you presented another periodic, if this was a complex tone, if you presented another complex tone of the same F0 40 milliseconds later, the person couldn't tell whether you started at the same phase that it would have had if it had really continued or not. So the brain doesn't seem to be conserving information about the phase or whereabouts you are in the cycle of the F0 over any kind of silent interval.
Yeah, I mean, one thing we haven't done in this experiment was follow up with some kind of behavioral or perceptual detailed analysis of what exactly this is used for. So right now it's just a physiological observation, and hopefully we'll be able to tell at some point, is this functional? Is this just kind of a spandrel, or is it something that's useful, and what are the perceptual effects?
Yeah, I mean, I can't really imagine anything, even like, any perception experiment. So for example, if you put in a little tiny 5 millisecond tone pip at different points, after that sound had been turned off, whether you would get any kind of, perceptually oscillatory behavior where thresholds went up or down, or the pitch changed, or you heard some kind of consistent change, cyclical change in the quality of the sound.
That would be really interesting. We're also thinking you could do some kind of gap detection here. Like if you started another stimulus on the same frequency, you could see if the next stimulus started here, you would expect that they wouldn't be able to detect a gap because it's still going on. But that's for future work.
They would have sort of done that with pure tones. And really the only ringing you get is the ringing you'd get on the basilar membrane, which is of a slightly shorter, rather shorter time periods. You don't really, we did experiments, which was mostly to do with FM, but we also did it with amplitude modulation, where if you take take an amplitude modulated tone and put a gap in it, and then fill that gap with a noise, then you hear the AM continue at a relatively low rates, like 5 or 10 hertz. But people can't tell when you reintroduce that tone after the noise if you started in the phase it would have had or the phase it wouldn't have had. People are completely deaf to that.
Oh,
even though you actually, so what we think people are doing both with FM and AM is they're kind of storing some information about the rate of the modulation and maybe the depth and the frequency, but not the phase. That gets thrown away relatively early in auditory processing. So you can't tell that phase discrepancy, even though if a sound really did continue and you had a sudden phase flip in it, then you'd hear that discontinuity.
Sorry for interrupting. I have a question to Bob that might be, Bob, is this effect of people not being aware, is that also revealed in, when they're listening passively in a kind of oddball type paradigm where you present a mismatching continuation versus a matching continuation? So
we've only done it behaviorally, basically. So you give them 3 intervals, 2 of which the stimulus continues afterwards with the preserved phase and 1 with the inverted phase, and see if they can tell the difference. There was a poster a million years ago by Daniel Presnitzer on the MEG, striatal continuity illusion, using the MEG to see if the phase of the stimulus was preserved over the duration of the illusion, and he found it wasn't. I was very interested in it, but he only ever did it as a poster. So.
Yeah, I think in that one they used a very specific kind of condition. But I mean, in your case, it may be that people are indeed unable to report, behaviorally. But it would be nice if there was some sort of passive listening type demonstration of.
Well, in a way, if you put electrodes in their auditory nerve, you'd be able to see that the phase was preserved, and you'd be able to pick the information out. But if you put electrodes in their finger, where they're pressing the response button, the information is lost. So it's somewhere between the auditory nerve and the finger, and I guess that's what neuroimaging is all about, isn't it? Really, just to find out where.
Yeah. At the risk of hogging the conversation, I don't want to prevent anybody else from asking. I have another question.
I no problems. I mean, there's a couple coming in the chat that you might see now.
Sorry. So why?
But we'll keep on grilling Emily forever. I mean, until her 6 o'clock. So it will be our midnight, if that's okay, Maria.
This is my favorite thing to talk about, so I'm in.
Good. Go for Hikaru's one, and then we continue with Maria. I have a follow-up for Bob, and probably related to Maria's first question, so I'll hold also.
Okay. So Hikaru in the chat says, have you checked the strength of the FFR to any features / region of interest of the structural MRI? So taxi drivers or expert musicians or professional ballet dancers have larger size and specialized brain regions. And I wonder if FFR also has a correlation with structural brain features. No, we haven't. The only thing we looked at is the white matter that's directly underneath the auditory cortex, and we found that there's a relationship between fractional anisotropy on the left side and the onset response. So we interpreted that as potentially some differences in white matter myelination, maybe carrying those quick impulses of sound up to the left auditory cortex more quickly. But that's really the only thing we've done with it. Yeah, I'm not sure exactly what you could quantify. Maybe you'd need some 7T for that as well, because these structures in the brainstem are extremely tiny. I don't know if we'd have the resolution to see small changes in their size.
Cool. Go for it, Maria.
Oh, I mean, it's a bit of a cheeky question and kind of related to Hikaru's question. So if the hypothesis is that that musicians might have stronger FFRs and so on, what about sleeping musicians?
Yeah. Okay, that's something we haven't looked at yet.
Because I mean, the question is, I mean, is it somehow related? So my entirely naive, I've never done FFR or sleep for that matter. But naively, I think, perhaps these differences are just related to vigilance or, commitment to the experiment, that somehow different groups of people are just, better at sitting and focusing. And that results in these differences that one sees. So perhaps sleep would be one way of controlling for that.
That's possible. I think that's a really important question also if you're thinking about potential clinical applications, because if your intervention requires some kind of top-down processing or constant attention, then it's not as good as a solution as if you can train that system to get better information all the time without you doing anything about it. We do have one small piece of data to put towards this question, which is when we recorded FFR to people doing other things and not paying attention to anything, they had a completely passive recording, we still see the relationship with musicianship and speech and noise processing. So that gives us some hope that it's actually something more burnt in, more kind of semi-permanent than something that requires online top-down control. It's probably both.
The worry, with passive listening is that it could still be the case that they are, somehow, trained or are more committed to sitting, to listening passively in some way without daydreaming about stuff in the same way.
I don't think we have the right sample for our sleep studies. We have, I think, 15 participants, and I don't think they will get enough of a range of musical training in there. But maybe something for the future would be interesting to look at.
Cool.
Were you our reviewer too?
Maybe I'm channeling them now, but no.
I have a quick related questions about, how you see this sort of relationship between state changes and cognitive changes, right? Because the state changes like sleep are so brutal, right? So global, so, and also so different between the states of sleep, that a really nice playground for I'm hitting the system with a brutal hammer or with an axe. So cognitively, I see it like a very strong thing, and that's from our previous discussions about it. While the cognitives are usually related more like a discussion or conversation between different cortical networks and so it's like smaller. But how you frame it, right? Because it's there will be a point where you can relate attentional aspects and prediction aspects to state aspects. Do you have a thought and a framework about it, or it's going to be coming later after the 200 monkeys in the house?
Yeah, probably later. I have a student who's really interested in comparing prediction within different levels of the auditory system, and he's going to be doing a 7T experiment and then MEG in the same people. Actually, the other way around. Otherwise, you magnetize people and you don't get anything in MEG. But we're hoping to be able to disassemble the system a little bit and look at that sort of thing. But I don't think we have any data that can speak meaningfully about that to date.
So I'm just sort of following on a bit from Maria's comments. So I think there is this issue always that when you're trying to pick an FFR, then if you have a lot of noise going on in the brain region at that frequency, then your component will be smaller. And it's really nice doing a training study in the sense that you overcome the being used to the sound, getting used to the experience versus the predisposition. But even in a training study, you can get non-specific effects. So the person just might be doing less overall cortical processing, or they might be more relaxed or more chilled. And I think quite a nice way to get around that is to have an a priori hypothesis that there is either some other sound presented at the time or some other component of the FFR which you don't expect to see change. And then you can say, look, you're looking for an interaction. So basically, you'll see a big difference between musicians and non-musicians on this component, and it will be significantly larger than the difference you see on the other component, or pre versus post training. And I think there was a little bit of that in a study by Carcagno and Plack ages ago, which did some training on, I can't even remember what type of task it was now. It was some kind of pitch task. So that would be another way of looking at it. But can I just make another comment about the FFR strength has got to be dissociated from musicianship in some way, because we know, for example, that the FFR is crucially dependent on age, and it decreases markedly above age 35. I remember when I started doing FFR experiments, I thought the equipment was broken, and the problem was they were using me as a subject. And then they got a 20-year-old, and oh yeah, it works. So what that means is that you're going to get an old musician with a tiny FFR and a young non-musician with a large FFR, but you don't really think that the type of processing which is going on in those, the old musician has somehow lost their ability to use, integrate with visual cues or do cross-modal grouping or any other things which you think that this FFR thing is measuring. There's got to be, like a huge amount of uncontrolled, not only just across subject variance, but even within subject over the same length of time.
Yeah, I'm actually surprised many of these behavioral results and correlations work out at all, considering the degree of variability that we see. Yeah. I find it really surprising how different they look.
Yeah. Well, another problem with the FF, in some studies, I'm sure not yours, but I think this is just a word of caution for people listening, because at the beginning you said, there's been extensive evidence showing that the FFR is affected by what you had for breakfast and the training session and, your social class and blah, blah, blah. And at least with a lot of those earlier studies, the problem is that the FFR has got a lot of potential dependent variables in it. So you can look at the phase lock coherence, you can look at the size of the component at F0 or at F1 or at F2, you can look at the left hemisphere, the right hemisphere. And a lot of those studies, even from the same lab, they'll use a different dependent variable in every paper. And there's this huge danger of double dipping. And with the musicianship equally, you can use dichotomy between musicians and non-musicians, or the age at which the person started training, or the number of years which they were training, or probably the square root of the difference between their age of there's a lot of stuff out there which is just really, is almost bound to be false positives. There's papers written on this. So that's where, I think that's where you are right when you say that replication is the answer. But a lot of these things aren't replicated, or at least with the same dependent variables.
Yeah, so this is an excellent point. This is an excellent point, and this is one that I first ran into when I started recording FFR. I was just looking for what had already been reported as a difference between musicians and non-musicians. Did not find it at all. So I went exploring, trying all these different variables, and then I was like, "What is this nonsense? We can't just pick the one we want." So I've stuck only with simple amplitude of the FFR fundamental for most of my studies. In the individual differences one, we use the phase locking value FO because Barbara Cunningham's student had said that seemed to be better for some reason, and then they changed their mind. So from now on, I'm only using the simplest.
You're going to stay pure to one measure,
yeah. But at least I can compare across my own studies.
Yeah, that's a start, isn't it? Which is puts you ahead of the game already.
Good. Final questions. I'll give you 1 minute before I close the session at 6. And before my wife keeps on waving at, Prosecco is ready, kind of thing. Any final questions? I'm reaching the final question sort of weapon.
Is this a Cambridge thing, Tristan?
I think I invented it the first Viva I did online, to be able to appoint someone. Oh, you finished your Viva, congratulations! And I did it with this, and then I started to use it for finishing talks. Final comments, or you're going to continue by email? I'm sure Maria has a list, of things to discuss.
I'm very happy to
drink.
Good. So I'm closing the question session. Thank you very much. And please join me in thank you, Maria, again, and Emily again.
Thank you very much for your attention and for your interesting questions and discussion. I hope to continue the discussion afterwards.
I forward you the questions I got in person.
Okay.
Thanks.
Okay.
Ciao.
Goodbye.